Hello, I'm Dr. Rohit, and today I'm going to discuss about enzyme immobilization. So let's first understand what do we mean by the term mobile and immobile. Mobile is something which can move. For example, this pen is mobile, it can move from one place to another. We human beings are mobile, we can move from one place to another. Plants, for example, are immobile, they cannot change their place. So immobile is something which cannot move, which are stationary at a particular place. Okay. What do we mean by the term enzyme immobilization? So it is a process where the enzymes, I have used this abbreviation for enzymes because this word is repeated again and again. So enzymes are physically confined, or you can say they are made stationary during a continuous catalytic process. Okay? So during our reaction, enzymes are used as catalyst. So what we do in this process, we make them stationary, and once the reaction is over, the enzymes may be recovered from the reaction mixture and they can be used over and over again. Okay, so this is what we mean by enzyme immobilization. Now, why we do this? This is done to improve the economy of the process. Okay, and how we do this? So this is achieved by fixing the enzyme molecules to or within some inert. Inert means a substance which does not react, and to a suitable material. So this is what, why, and how of enzyme immobilization. What means, what is this process? Why we do this process? This is one of the reasons. There are many reasons which we are going to cover in the next few minutes. Okay, and then how we do this. Okay, the definition of enzyme immobilization as per your textbooks. So enzyme immobilization, the short form I've used is EI. So it refers to enzymes which are physically confined, means they are stationary, they are localized, where in a certain defined region of space, means they are localized or they are stationary in a defined medium or a defined material. For example, in some hollow fibers, or they are linked to some fibers, or they are linked to membranes, which we are going to discuss, with retention of their catalytic activity. Means this process does not affect the catalytic activity of the enzymes. They are fully functional even after they are immobilized. Okay, and these enzymes which can be used repeatedly and continuously. So in this way they save a lot of money, they are very economical as per the industry requirements. Okay, let's quickly see why we need enzyme immobilization, or why this process is very important from the industrial aspect. So the first reason is it improves the economics of biocatalytic process. Why? Because you can use the enzymes again and again. Okay, so it reduces the cost of the overall process. Second, it allows the enzyme to be stationary throughout the process. Enzyme is stationary. Enzyme is immobile throughout the process or throughout the reaction. So once the reaction is over, we can easily separate the enzyme from the product, and also the product is not contaminated from the enzymes. For example, if the enzymes are not confined to a certain space, and they are in a reaction mixture, it becomes difficult to separate them from the product. But if the enzymes are confined to a stationary medium, or if the enzymes are immobile, it becomes very easy to separate them from the product, and the product is also not contaminated from the enzyme. For example, enhance the stability. So now enzyme immobilization, it also enhance the stability of the enzymes. Okay, enzyme immobilization improves many properties of enzymes, such as the performance in organic solvents. It improves the pH tolerance of the enzymes, it improves the heat stability, and at times it also improves the functional stability of the enzymes. Okay, so that is why this process is very important. And last, it gives you the ability to stop the reaction by removing the enzyme from the reaction solution. For example, if you are making the tea with the use of tea bag, as soon as you got the desired color of your tea, you can easily take out the tea bag and the reaction is stopped. So in the similar way, you take out the immobilized enzymes from the reaction, and the reaction will stop. Okay, let's understand the role of a good support or carrier which is used in the immobilization process. Okay, so the main function of the support or carrier is to stabilize the structure of enzyme. Okay, and thereby preserving the efficacy of the enzyme so that they are more resistant to the surrounding mediums. Okay, so this is the role of an ideal carrier or support. Now let's discuss the properties which we should consider while selecting an ideal support for a particular type of immobilization process. Okay, so the first property is the physical property, and what are the points which we need to consider? That it should have the sufficient strength, it should have the sufficient surface area or the large or the desired surface area. Then it should have a good pore size. The pore size is important because it should be in accordance with the size of the enzyme. If the pore size is too big of the support system, the enzyme will go inside the support and it will not be available for the reaction. And then the density of the carrier, and then the physical form in which the carrier or the support is going to be used, whether it is going to be used in the form of beads or in the form of film or in the form of fibers. Okay, and second is the chemical property. What points we should consider? That the carrier should be hydrophilic, it should have functional group for modification. This particular point we are going to discuss in the next session, where we will discuss the types or the methods of enzyme immobilization. Okay, and then it should be inert towards the enzyme. The carrier or the support should not react with the enzyme. Okay, and it should be reusable. Property, which is the stability. In this, the points needs to be considered are regeneration of enzyme activity, which means once the reaction is over, you have to use the enzyme for the next reaction. The activity of the enzyme should be readily regenerated. How? By supplying them with the necessary cofactors or coenzymes, and then the maintenance of enzyme availability. It should not happen that with every reaction we are losing the number of enzymes. Okay, this should not happen. And the stability of carrier or support is very important to maintain the enzyme availability. Okay, and third is the and then is mechanical stability, which means that any wear and tear during the process should not affect the stability of the carrier. And fourth is the resistance to microbial attacks, resistant to temperature changes, resistant to change in the pH, and resistant to proteases. As you know, enzymes are proteins in nature, so different proteases can damage the structure of the enzyme. Okay, so the support should be resistant to all these factors. And some okay, and there are some miscellaneous factors which needs to be considered, which are safe for process workers and end product users. Okay, so it should be safe for the workers who are immobilizing the enzyme on the support, and once the enzyme has been immobilized, so the people who are going to use that immobilized, it should be safe for both the category of people. And then the cost of the carrier is very important. It should not be very expensive. Availability, it should be readily available. And the last is feasibility for scale up, which means that with a particular support or carrier, it should be possible to use them on a large scale and not just on the laboratory scale. Okay, we have come to the end of today's discussion. I hope you really enjoyed the segment. Thank you so much.